Welcome to this comprehensive exploration of small intestine absorption and transport. The small intestine is a remarkable organ where the final stages of digestion meet the critical process of nutrient absorption. In this presentation, we'll examine the intricate mechanisms that allow our bodies to extract and utilize nutrients from the food we consume. Let's begin by understanding the fundamental transition from digestion to absorption. While digestion breaks down complex food molecules into simpler forms, absorption is the process by which these nutrients cross the intestinal barrier and enter our bloodstream or lymphatic system. Both processes are essential, but they serve distinctly different roles in nourishing our bodies. The structural foundation of absorption lies in the villus. Each villus contains a central lacteal vessel surrounded by blood capillaries, all enclosed within an epithelial lining. This elegant architecture is perfectly designed to facilitate efficient nutrient uptake and transport. One of the most impressive features of the small intestine is how it maximizes surface area for absorption. Through the presence of villi and microvilli, the small intestine creates an enormous surface area within a compact space. This structural adaptation dramatically enhances the efficiency of nutrient absorption. The brush border formed by microvilli serves three critical functions. First, it provides a structural advantage by increasing surface area. Second, it acts as a transport hub, housing numerous carrier proteins and enzymes. Third, it serves as a protective barrier, helping to regulate what enters our body. Water-soluble nutrients follow the portal route. Monosaccharides, amino acids, water-soluble vitamins, and minerals are absorbed into blood capillaries and transported directly to the liver via the hepatic portal vein. The liver then processes these nutrients before they enter systemic circulation. Fats take an alternative route through the lymphatic system. Dietary fats and fat-soluble vitamins are packaged into chylomicrons and absorbed into lacteals. These nutrients bypass the liver initially, entering the bloodstream through the thoracic duct. Comparing these two absorption routes reveals important differences. Portal circulation offers rapid absorption and immediate liver processing, making it ideal for water-soluble nutrients. Lymphatic circulation provides a slower route that's essential for fat transport and allows fats to bypass initial liver metabolism. The gut barrier plays a critical role in protecting our body. It must selectively allow nutrients to pass while blocking harmful substances, pathogens, and toxins. This selective permeability is maintained through sophisticated cellular mechanisms and immune defenses. Tight junctions are the key to barrier function. These zipper-like proteins seal the spaces between intestinal cells, forcing substances to take the transcellular route through cells rather than the paracellular route between cells. This control mechanism is essential for maintaining barrier integrity. When tight junctions become compromised, intestinal permeability increases. This condition, often called leaky gut, can allow unwanted substances to cross the intestinal barrier, potentially triggering inflammation and immune responses. Various factors, including diet, stress, medications, and infections, can affect tight junction integrity. Maintaining barrier integrity requires multiple factors working in harmony. Nutritional support through adequate protein, vitamins, and minerals is essential. A balanced microbiome produces beneficial compounds that strengthen the barrier. Lifestyle factors like stress management and adequate sleep play important roles. Finally, proper immune regulation helps maintain the delicate balance between tolerance and defense. Nutrients cross the intestinal barrier through two main mechanisms, active and passive transport. Active transport requires energy and can move nutrients against concentration gradients, making it essential for absorbing nutrients that are present in low concentrations. Passive transport relies on concentration gradients and requires no energy expenditure. Carbohydrate absorption demonstrates these principles beautifully. Glucose and galactose are absorbed through active transport using the sodium-glucose cotransporter. Fructose, however, uses facilitated diffusion, a form of passive transport. These mechanisms ensure efficient uptake of all dietary sugars. Protein absorption involves 5 key stages. First, proteins are broken down into amino acids and small peptides. Second, these products are transported across the brush border. Third, any remaining peptides are broken down inside the cell. Fourth, amino acids are released into the bloodstream. Finally, these amino acids are delivered to tissues throughout the body. Fat absorption is more complex due to the hydrophobic nature of lipids. Dietary fats are emulsified by bile, broken down by lipases, and absorbed as fatty acids and monoglycerides. Inside intestinal cells, they're reassembled into triglycerides and packaged with fat-soluble vitamins A, D, E, and K into chylomicrons for transport. Bile plays an indispensable role in fat absorption. Bile salts, synthesized in the liver from cholesterol, emulsify fats to increase their surface area for enzyme action. These bile salts are then recycled through enterohepatic circulation, making the process highly efficient. Disruptions in bile production or secretion can significantly impair fat digestion and absorption. Looking ahead to clinical connections, we'll explore how understanding these absorption mechanisms relates to real-world health issues. We'll examine malabsorption syndromes and their nutritional implications, investigate the relationship between gut health and systemic disease, and discuss therapeutic strategies for optimizing nutrient absorption in clinical practice. Let's summarize the key concepts we've covered. The small intestine structure, from villi to tight junctions, is exquisitely designed for its function. Multiple absorption routes and mechanisms ensure efficient nutrient uptake. The gut barrier protects us while allowing essential nutrients to pass. Understanding these processes is fundamental to comprehending human nutrition and health. In conclusion, small intestine absorption and transport represents one of the most sophisticated biological systems in the human body. The structural adaptations that maximize surface area, the selective barrier function that protects while permitting nutrient passage, the diverse routes and mechanisms for different nutrient types, and the clinical relevance of these processes all underscore the importance of intestinal health. A thorough understanding of these mechanisms is essential for anyone studying nutrition, medicine, or human physiology.